Okay, hello everybody. Thanks for joining to this virtual EPR seminar. So you should have probably gotten a notification now that this is recorded. So if you don't want to be on this recording, just don't unmute your video and your sound and you will not be on it. Then also please just mute yourself during the talk because background noises can be quite disturbing. And would be easiest if you can manage yourself and it's not me that has to do it. I think that's it for the boring part. So today we have Joshua Wort from St. Andrews. He did his bachelor and master's in biochemistry at UCL with Chris Kay, actually on HSP 90. He did a brief internship at CNRS in France, and then he joined Bela Bode in St. Andrews for his PhD, where he's concentrating on method development. And today he's talking about optimizing copper-2 chelate labeling at double-histidine motifs for pulse dipolar EPR applications. Josh, the stage is yours.
All right, thank you very much, Nino. Thank you everyone for coming. So as Nino says, I'm going to talk today about optimizing copper-2 chelate labeling at double-histidine motifs for pulse dipolar EPR applications. But before I get into the details of this, I just thought I would give an example for how to motivate or rationalize our group's interest in these double-histidine motif-based spin labels. So here I show a DNA helicase protein. This is XPD. And you can see in the black box here, this is an iron-sulfur cluster, which is coordinated by several essential cysteine residues. Now, one of the main appeals of these copper-based spin labels is that they're chemically orthogonal to nitroxide-based radicals, or really any cysteine-directed spin label. And actually, you can spin label XPD with MTSL, but your labeling efficiency is not great, and you also have to worry about whether or not you're perturbing the structure here. So when you introduce your double histidine motifs in a pair, you can coordinate your copper label and then measure your time trace here, which I've shown at the right. And as the inset, you see that we get a nice distance distribution. It's bimodal here, and this is the other point to make about these spin labels, that they confer very nice precision in the distance domain. So I'll touch on this in a bit more detail momentarily, but XPD also has a subtle conformational change, and so we're sort of actively pursuing the source of this splitting in our group. So the double histidine motif was used in CW EPR 2 decades ago by Hubble, but it wasn't appropriated for pulse dipolar EPR spin labeling applications until reasonably recently by the Saxena lab. And the motifs typically come in 2 configurations. So you can have your histidine residues arrayed at positions I to I+2 along a singular beta strand of a beta sheet secondary structure, or at positions I to I+4 along an alpha helical secondary structure. The copper labels themselves are typically introduced as chelates, and the function of these chelating agents is to confer a very high degree of specificity to prevent off-site interactions at other positions in your protein. Now, I show the model protein GB1 here. This is the B1 domain of protein G of Streptococcus G, and this was used in the initial study by Cunningham et al., but I show it here and throughout the presentation because we also use it as a model system for the benchmarking work that comes later. So I've already touched on the fact that these labels are appealing because they're chemically orthogonal to nitroxides, for example. But another advantage is that they self-assemble, so they're genetically encoded, and this self-assemblage occurs on a reasonably short timescale. Now, they're also modular in their complexity, which means you can alter the configuration of the histidine residues that you're using or the nature of your chelating agent. But as I touched on in the previous slide, the main advantage of these spin labels is that they confer exquisite precision in the distance domain. And to emphasize this point, I've just shown bis MTSL GB1 in the top here, and the tetrahistidine GB1 construct in the bottom, with some simulated distance distributions in red and blue respectively. And what you can see is that the tetrahistidine case affords a much narrower distance distribution. And this comes from the fact that the copper center has a much reduced rotameric freedom with respect to the alpha carbon of the protein backbone. And this is relative, for example, to MTSL. And just to further emphasize this point, that makes these labels particularly useful in the study of systems that have subtle conformational changes or subtle conformational equilibria, which otherwise you would be insensitive to. So because the spin labeling strategy is a coordination-based approach, it does mean that it's potentially vulnerable to low binding affinity. Certainly the initial studies reported binding affinities in the low millimolar concentration regime. And so we were interested to try and reproduce this to independently investigate each of these double histidine motifs. And just to clarify here, when we talk about the binding affinity, we can quantify this for dilute solutions according to the expression given here in the green box. Now, in order to investigate the affinities of these motifs, we engineered a pair of novel GB1 constructs which contained cysteine residue for MTSL labeling and one of each of the double histidine motifs on the opposing face of the protein. And to do this robustly, we used a combinative approach of isothermal titration calorimetry, which is a sort of golden standard for the thermodynamic characterization of protein-ligand interactions, and also the 5-pulse dead time-free RIDME experiment, the sequence of which I've shown here for completeness. So another thing to note about these copper-based spin labels is that when you are at Q-band or higher frequencies, typically they're spectroscopically orthogonal with respect to, certainly with respect to nitroxide radicals, but also other organic radicals. And that means that you can detect each species very selectively. Now, I emphasize this point here because it has implications for the subsequent slides. But if we consider just detecting on the nitroxide spectrum in the first case, then our modulation depth will be hyperbolic with respect to increasing copper-2 concentration. This is because the excess copper doesn't contribute to our background, and therefore it won't wash out the modulated signal. If instead we move to the maximum of the copper chelate spectrum, then we see that our modulation depth profile is log normal with respect to increasing copper-2 label, because as you obviously know, then in that case, the copper chelate will contribute to our background, and this will detriment our sensitivity at higher excesses. So typically we performed these RIDME pseudo-titration series by nominally detecting on the nitroxide, and then systematically increasing the concentration of our copper chelate within our system, so that the intramolecularly bound copper-2 would modulate the detected nitroxide, and this would lead to our modulation depth reporting the fractional saturation of our double histidine motif. And I've shown that schematically in the top here, but also at the bottom left I show an exemplary isotherm. We see that it has the hyperbolic shape that I mentioned previously with increasing copper chelate concentration. And this is reminiscent of a Langmuir isotherm, which comes from, or you would expect for a singular binding site. I also show the equations that we use here to fit this curve. I'm not going into really any detail about that now, but if you have any questions about that, please feel free to ask at the end. So just to now direct your attention to the experimental RIDME pseudo-titration series, we performed several of these, but I only show one here. This is significant for a couple of reasons. So this is the alpha helical double histidine motif measured in presence of copper NTA. And each of these measurements were acquired overnight and performed at 500 nanomolar protein concentration. So serendipitously, we found that we had an exquisite concentration sensitivity afforded by copper nitroxide RIDME. I should mention that we didn't initially start measuring at this protein concentration. Rather, we measured at much higher protein concentrations, but realized that we couldn't resolve the curvature of this hyperbolic function, and therefore had to systematically reduce it. When we fit a KD to this saturation function, we found that we had a KD of 140 nanomolar. So the affinity was obviously much higher than was previously speculated. And this was at odds with the previous findings. So just to direct your attention briefly to the central panel here, we can use the corresponding distance distributions as a sort of internal control to confirm that we have specific and directed binding at the double histidine motifs. So this tells us that we don't have any non-specific interactions that would otherwise contribute to the isotherm. And that's exemplified by the fact that the only specific feature, the only significant feature in here is this main peak at 2.5 nanometers. So I mentioned that we wanted to use ITC in order to validate this, this observation. And so we performed ITC at room temperature for all copper chelate and construct permutations. So there are a couple of key points to mention. I showed the raw data and the corresponding fits here. But all of the KD values were predicted to be in the low micromolar concentration regime. And the nature of the binding was consistently observed to be exothermic. So that means that the binding affinity would increase with decreasing temperature. And this would be in line with the observation of a high nanomolar KD under cryogenic temperature conditions that we observed with our RIDME pseudo-titration series. So with this in mind, we wanted to see if we could extrapolate the ITC data to cryogenic temperature, because if the RIDME determined KD value is representative and accurate, we should in principle be able to reconcile the two techniques. So to do this, we used the Van Hoff equation, which I've just shown at the top here. And when we did this, we found that actually the EPR and ITC determined KD values agreed reasonably nicely in quite a narrow temperature range. We see from the crossing points here that I've marked in black that this is actually quite reproducible between series. And what this tells you is that actually the RIDME isotherm is representative of your binding equilibrium at this temperature. And at this temperature, your cooling rate exceeds your translational and diffusional processes, which would otherwise contribute to your equilibrium, which at this temperature and below begin to freeze out. So that was quite nice that we were able to reconcile these two approaches. But we were also able to show that the steepest error surface for the pseudo-titration series was observed under conditions of 500 nanomolar protein concentration. And this comes from the conventional wisdom that says if you want to measure a low KD value, one should measure in a correspondingly low protein concentration regime. So just to round up the first part of the talk, we were able to show that copper nitroxide RIDME afforded exquisite concentration sensitivity. We were able to show that we could use RIDME to investigate binding equilibria and reconcile the determined KD value with ITC data using the Van Hoff equation. And we know from the previous work of the Sexina lab that these spin labels confer very high precision in the distance domain. So we were also quite surprised to note that the copper-copper RIDME had an equivalent concentration sensitivity. And this then led us to ask the question, is it possible for us to extend this approach so that we can apply it to more complicated binding equilibria in the absence of a nitroxide-based spin label? And this has actually some interesting implications, because if we think about the tetrahistidine GB1 here as an example, how is one to best optimize the labeling efficiency of this construct? If you have very high binding affinity, this is a reasonably trivial problem. But under conditions of lower binding affinity, that is, if your labeling is non-quantitative, you'll need an excess of your copper chelate in order to saturate the site. But as I mentioned already, you don't want to add a vast excess. And so it's about finding the right balance. So to do this, we essentially derived a general speciation model for a pair of KD values and a given protein concentration. And then what you can do is essentially input these values for a vector of ligand concentrations. And on the right here, I've shown the modulation depth profiles as a function of the copper label concentration. And you see that you can essentially optimize your sensitivity. So if you look at these curves, the significant point is the sensitivity optimum is a point solution on the curve. So it isn't a plateau anymore. It's not hyperbolic. It's log normal. And that's why this is potentially of interest, not only for copper double-histidine motif spin labeling, but also any coordination-based spin labeling approach. So we already had the tetrahistidine GB1 construct as a gift from Sunil, and therefore we endeavored to perform another RIDME pseudo-titration series in presence of copper NTA. So using the previously determined KD values from both the ITC and copper nitroxide RIDME, we were able to simulate a modulation depth profile, which I've shown in the black here. And you can see that the experimental modulation depths follow this pattern reasonably well. Only under conditions of high concentrations do we see deviations. This is probably because of non-specific binding interactions. We also see from the corresponding distance distributions that we do have some artifact peaks which survive validation. So that does seem to suggest we have non-specific binding, but I'll touch on this later on. If we first look at the dipolar evolution function in the middle panel here, the magenta case, you are very close to the sensitivity optimum, and we have the best modulation to noise ratio. We also have the best or the cleanest distance distribution. And this really provides a rationale for why one would want to optimize this labeling efficiency, because we see under conditions of under-labeling, the distance distributions are really quite nasty. And even under conditions of over-labeling, we have artifactual peaks which simply don't appear in the optimally labeled case. But perhaps the sort of the coolest aspect of this approach is that in principle we can extract a pair of KD values from a singular RIDME pseudo-titration series. And we were quite surprised because we found that we had very nice agreement with the previously determined copper nitroxide RIDME values. And we also saw that the KD values were not compensatory. So you might expect that as one KD value increased, the other one could decrease and therefore kind of compensate the fit. But that's not the case. It doesn't manifest that way experimentally. And this has implications for the robustness or the uniqueness of these fit solutions. Just as a sort of sanity check, we wanted to do a global fitting of both the copper-copper and copper nitroxide RIDME to see that the results were consistent, and indeed we found very nice agreement for both series. So this is just a nice sanity check that our model is behaving as we would expect. So just to change pace slightly at the end here, we've started to investigate the influence of environmental factors such as pH and also competitor metal ions on double histidine motif affinity using room temperature ITC. So just to begin with the pH series, we started measuring under acidic conditions, and we found that, as you might expect, under pH 5 conditions, we could completely ablate double histidine motif binding. And at pH 6.4, we observed a 20-fold reduction in binding affinity. This is because the histidine residues are obviously protonated and therefore cannot, the copper chelate cannot properly coordinate. If we then move to pH 7.4, we find a recovery of binding. So we get a KD in the low micromolar regime again. And then at pH 8.4, this is quite interesting because thermodynamically, this binding is actually much stronger than at pH 7.4. But you also have this increase in the stoichiometry here, which suggests that maybe under high pH conditions, you begin to deprotonate the rest of your protein surface. And therefore, this might lead to non-specific binding interactions. So this is something that we're sort of actively pursuing, but the larger implication for this is that actually this affinity of binding is tunable, especially for acidic conditions, but as well, you see that there may be some saddle point or optimal pH where you really want to measure and to optimize your labeling efficiency. So we also could show that nickel NTA and zinc NTA could bind double histidine motifs at room temperature. And they do this, as you would expect, much weaker than copper NTA does coordinate double histidine motifs. But what's interesting about this is that you can use, in principle, we have mathematical models that essentially mean we can use competition assays to indirectly determine the binding affinity of diamagnetic species. So this curve on the right here, this pair of curves on the right, this is a kind of competition pseudo-titration. And this assumes that your competitor ligand is diamagnetic, which obviously for nickel NTA is not well met, but for zinc NTA this is the case. And this is something that we're actively looking into at the moment. So in conclusion, I hope that I've shown that not only are double histidine motifs great for spin labeling applications, but they're also very nice model systems that are simple and modular for the study of binding equilibria by pulse EPR, by dipolar pulse EPR. So we were able to show that we could benchmark the previously the previous work using copper nitroxide RIDME for a simple one-site binding model, and extend that to a two-site binding model using copper-copper RIDME. We found previous KD values from both ITC and copper nitroxide RIDME to be in excellent agreement with our empirical observations. And we started to investigate the influence of environmental factors such as pH and competitor ions. So in the future, we want to see if we can recapitulate the reduction in KD under acidic conditions using another copper nitroxide RIDME pseudo-titration. We want to see if we can indirectly determine the affinity of binding of zinc NTA using a RIDME competition assay. And we've started to expand our speciation model to include cooperativity and non-specific binding constraints. So with that, I'll just finish off. And I want to thank especially Bella, but also the rest of the Bode lab, Angeliki for her help with benchmarking the synthetic model systems that predate this work, David Norman, my second supervisor at the University of Dundee, Doctors Alan Stewart and Swadia Arya at the School of Medicine at St Andrews, and the mass spectrometry and EPR facilities at St Andrews, the funding bodies, and of course you for your kind attention. Thank you.
You can't hear them, but I hope my applause was representative. Thanks, Josh. Now we have time for questions. So you should be able to raise your hand digitally. I think it's under participants or something like this. I don't have it because I'm a host. Let's start with Maxime.
Yeah, very nice presentation and also nice and useful work. Congratulations. I wanted to ask, it was not clear to me when I saw this kind of presentation before, that actually at ambient temperature, when you're performing the labeling, the KD is not as good as it is at low temperature. So this is one of the points, right? Regarding this, can you maybe still comment a bit more, kind of a bit more extended, what precautions one would then need to use this labeling if it is, let's say, compatible with, I don't know, nucleic acids? What would you see, where, if and where you would see any limitations that one should avoid or kind of carefully check when using these copper NTA or ID labels?
I think it's a really good point. So just perhaps I start more generally. So I mean, the beauty of these labels is that the binding is exothermic. So if the binding were not exothermic, and it isn't always exothermic, that there are other protein ligand interactions that are obviously endothermic, then in that case, study with pulse EPR is probably limiting, assuming that your sort of the kinetics of your association are very fast with respect to the rate of cooling. So, I mean, what we found was that essentially, and something we were eager to sort of look into in more detail was the composition of the matrix. So I suspect this would actually be something that you could modulate if your binding affinity was not very good, even at a given temperature. If your freezing point temperature and the glass transition temperature of your matrix were low enough, assuming you had an exothermic interaction, you could drive that association to completion, if that makes sense.
Yeah. No, I mean, it's clear that if you have a mixture, even though at room temperature, the association is not completed, then you cool down slow enough, and then you get it almost 100% complete. But then this means that at room temperature, many things can happen. For instance, you need to make another purification step, then of course you're losing the copper label, right?
Yes.
Or you have something else which could bind, and then even if it is binding with lower affinity, this might be like, I don't know, in excess or whatever. If you, at room temperature, if you have a dynamic balance, like that was the core of my question. So what obstacles you might see due to this dynamic balance at ambient temperature before you cool down?
No, so I think you are completely right, and that is a problem with this approach. That you are right that your label would probably fall off. So really, the spin labeling is the last thing that we do, and I can't really comment on how effectively it sticks around if you label prior to, for example, needing to do additional purifications and such, but I would imagine that the label falls off pretty quickly. So ideally, you would do the spin labeling as the last thing, and kind of troubleshoot everything else before getting to that step.
I think there is no approach without difficulties, and this is nevertheless a very good one. It's just, I think it would be interesting to know, at least for some key biomolecules, what interferences can happen for such systems. I think that's just an interesting question.
Okay. Then let's move on. I mean, we can come back to that point, Max. Kendrick?
Yeah.
Kendrick? You have to unmute yourself if you want to.
Yes. Hi, how you doing? Very interesting talk. I wanna ask. Say you're saying that this copper motif is very specific to a double histidine motif, but the question is, does it depend on the local structure of the protein? Say when it's expanded or when it's in a coil. Does that affect the binding of the copper to the histidine motif? And the second question is, have you done any study to see how well these copper partition into different hydrophilic or hydrophobic environments?
Yep. So maybe I start with the first question. So that's a really good point. I think, I mean, I can only speculate because to my mind there aren't enough generic widespread applications for this that would paint a definitive picture of having, say, high affinity for all double histidine motifs on a helix or all double histidine motifs on a sheet. I would be very surprised if the surrounding protein structure didn't play a role, but this is something that I think just needs more investigation. So at the moment, this is kind of speculative, but at least for the in the majority of the proteins that this has been applied, they have observed reasonably high binding affinities. This is in a number of different systems where they have different binding environments in their secondary structure. So the second question, sorry, if you could just repeat that for me again, that would be great.
The second question was, have you done any study to see how well the copper spin label partitioned into environments of different
No, we haven't yet.
Sure. Thank you very much.
Okay, then Thomas.
Hey, thank you, Joshua. Great talk. Quick question. In solution NMR, when we measure normally PREs, we on average go over around 5 spin labels, gadolinium, manganese, nitroxides, just to see what are the non-specific binding occurring. Like non-specific meaning like labeled to protein randomly, like protein to protein. Now, you hinted towards it that you did the validation to detect some distances which are rising for non-specific interactions. But considering that normally we get already a whole bunch of distance here and there, which are well, one time can be due to noise and God knows what. What's a good approach to determine? Let's say you go through 5 different metals, and you want to determine which one is the best. What's probably the best approach for that?
Well, with respect to the specificity of binding, I mean, I'm not sure if there is a kind of a rule of thumb that one could use. I mean, this is also particular for the model system that we are using. So yeah, it will vary from system to system, but I think, I mean, if I just draw your attention to, if you were talking about using this kind of approach here, you can see that, I mean, you can speculate that you have some non-specific binding, as you say, but really and we did this for the previous copper nitroxide RIDME pseudotitration series, that we in that case detected copper-copper RIDME. I'm not sure if that answers your question, but I think it comes down to how easy it is for you to build a chelator for that metal, and then whether or not that would perturb your structure.
Right. Well, yes. Okay. All right. Thanks.
Then Ilya, please.
Hi, nice talk. So a bit of a serious here, maybe a silly question. If memory serves, copper binds to the nitrogen of tryptophan, to arginine, and to quite a few, other side chains that contain nitrogen.
Yes.
So as a matter of safety, would it be a requirement that a protein doesn't contain other histidines, arginines, and such like? Because if memory serves, the proteins are held together by literally the equivalent of a few hydrogen bonds in energy, and now you introduce something that strongly coordinates. Can it completely destroy the protein if there are too many?
I think this is really the function of the chelating agent. So the way we normally do the spin labeling is that we introduce the copper that's already chelated. So NTA is tetradentate, I think, and therefore the copper has only the two.
I see. So you are saturating other coordination locations.
Yes.
This. Okay.
Yeah. So you are completely right that I think that would happen if you had copper without your chelator, but in this case, you get around it. Yeah.
Clever.
Okay. Then Angeliki.
Hi, Josh. Thanks for the nice talk. I want to ask you about the copper-copper README. Maybe if you can go there with the KDs. So yes, like this slide and the previous one you showed, maybe. So basically here you said that we have the extra distances in the distance distributions because we have some probably unspecific binding of the copper, right?
Yes.
And this is more pronounced probably at the higher concentrations, right? But from what I see here, the purple trace, which is the top one, is the cleanest one. So I see the extra peaks coming at the low copper NTA concentrations, the 1570 micromolar. Is it just a mixed up of colors, or is it something I don't know?
No, I think so. No, I think you have a point, but essentially the.
So basically, the red and the dark blue, the two lowest distance distributions.
Yeah, I mean these ones will be these ones will be corrupted with noise. So
okay, but these are the ones with big excess of copper.
No, the two lower ones are the ones with under labeling.
Under labeling. Okay.
Yeah, these have under labeling, and then the orange on the top and the green, these have an excess.
And then the small kind of peaks, you say these are the ones corresponding to one specific binding.
Yeah, I mean you see for the green trace here that you have some
right.
At 3 and some at 2 here. I mean, this is just speculative. We don't know for sure that it's non-specific binding, but
yeah.
I mean, I know that Sinil observed that there was this secondary binding site for copper in GB1, so like a native copper binding site. And so then the question is, if you have such a vast excess of copper NTA that, I mean, the off rate won't be very high, but if it's enough, then maybe you begin to saturate this site.
Yeah. And one more question here. So basically for the 500 and the 1000 micromolar copper NTA, maybe we have some unspecific binding and so on. my question is there, how did the T1 profile look like? Was it similar? Like, did you have to adjust the mixing time?
So that's actually a really good point, and this comes back to what I said here about sorry. Let me just find this. Yeah, here we go. So basically, this comes back to the equations here. So the point is, you are right that your T1 will nominally it would increase with increasing concentration, right? And so you need to essentially normalize away that effect. So to do that, we don't take the raw modulation depth, but we take the quotient of the observed modulation depth and the asymptotic limit of the modulation depth for a given ratio of mixing time and T1.
Okay, I see.
Okay.
Okay.
And then
okay,
you essentially remove the effect of increasing your of decreasing your T1 sort of artificially with concentration. Yeah.
Like in this case, would it just make sense to keep a constant mixing time, no matter what the T1 looks like for the different samples, even if the absolute value of the mixing time is not correct, but then you can say from one ratio to the other that you really see the species you want.
In the case of the copper nitroxide, I think it is necessary to...
Not for the copper nitroxide, for the copper-copper.
For the copper-copper, we basically... so I think what's relevant is to keep a constant ratio between your T1 and your mixing time, but not necessarily a fixed T-mix value.
Yeah. Okay, makes sense. Thank you.
Okay, other questions? Thomas, I see you raising your hand. You can ask.
Quick question. Just thinking about what what's her name, Angelica said. What if you do the DO experiment? What you did was a different concentration, and to determine like how many coppers are bound, you can do just simply vary the flip angles. I don't know how the rhythm necessarily works, but in similar in DO, you can pretty much just vary the power of the flip angles. And then the modulation depth should vary as a function of how many coppers are present in the vicinity of the evolution time. Meaning that you could actually get not necessarily an accurate measure like how many coppers are there, but this would actually help you to pin down if there's how much non-specific interaction between your metal and your protein occurs, without dealing much of like isotope labeling or any rubbish like that. So actually I think that might be a really good approach to determine, let's say you have 5 metals, you test 5, you do each one, you just do a series of like 5 flip angles at some specific concentration, and then you can fit it. Obviously your gases lab has done a lot of that stuff. To an equation, and then get how much non-specific interaction you have for each individual metal.
Yeah, so I think I think that's a really good point. And actually, we are looking at trying to use kind of a combinative approach of Peldor and RIDME together in this, but I can't say too much about that at the moment because it's all sort of preliminary, but yeah.
Also, Thomas, maybe to the different metals. I mean, you cannot just take any metal. I mean, there are always other complications coming. So if you have manganese, I have like these overtones and stuff. If you do RIDME, yeah. So, yeah.
Of course, yes.
Any other questions? It doesn't seem to be the case. This is not, thanks Josh again, I do it like
Thank you.
For everybody, and stop the recording.